Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
The genetic code that dictates how genetic information is translated into specific proteins is less rigid than scientists have long assumed, according to research published today, November 9, in eLife. In the paper, scientists report screening the genomes of more than 250,000 species of bacteria and archaea and finding 5 organisms that rely on an alternate genetic code, signifying branches in evolutionary history that haven't been fully explained. To perform this synthesis, ribosomes read strands of mRNA, copies of bits of the organism's genome, in chunks of 3 bases at a time. Each 3-base sequence, known as a codon, binds to a specific transfer RNA, tRNA, that ferries a corresponding amino acid to the ribosome to be added to the protein chain. An organism with an alternate genetic code, like the 5 new instances that the study authors found, has codons that correspond to different amino acids than they would in the standard genetic code employed by the vast majority of known life forms. The genetic code has been set in stone for 3 billion years, study co-author Yekaterina Shulgina, a Harvard University graduate student in systems biology, tells The Scientist. The fact that some organisms have found a way to change it is really fascinating to me. Changing the genetic code requires changing ancient, important molecules like tRNAs that are so fundamental to how biology works. As such, the code was thought to be largely preserved across all forms of life, with scientists finding the occasional exception during the past several decades of RES. In addition to finding 5 new alternate genetic codes, the team also verified 7 others that had been discovered one by one in the past, bringing the total number of known exceptions in bacteria to 12. I'm pleased to see that all of the results that we had so far came out in the new paper. Yale University biochemist Dieter Saul, who didn't work on the study, tells The Scientist. Saul has been studying the evolution of the genetic code for decades and was the first to find an alternate genetic code in bacteria. The team's methodology was very good, he says, especially because of how it illustrates that the genetic code is exceedingly flexible. To re-find everything that we already knew and to double the number of known reassignments was particularly impressive, says University College Dublin Genomic. What they're looking for is really rare. Shulgina and study co-author Sean Eddy, a biologist at Harvard and a Howard Hughes Medical Institute investigator, developed an algorithm called CODETA, named after the Rosetta Stone, that can screen an organism's genome and predict which amino acids it's cod. From there, researchers can experimentally validate its work by looking for the predicted tRNA in the organism. CODETA excels at finding genetic codes that are nearly the same as the standard. Prior to the new study, all known alternate genetic codes in bacteria involved changing a stop codon that instructs cellular machinery that it's reached the end of a protein into one associated with an amino acid. The new results reveal the first alternate genetic codes in bacteria that represent a sense codon reassignment. That is, changing a codon from one amino acid to another. Accounting for deviations, then, will improve the accuracy of those predictions and prevent errors from being codified into databases as more and more genomes are sequenced. Changing the genetic code requires changing ancient, important molecules like tRNAs that are so fundamental to how biology works. Yekaterina Shulgina, Harvard University. But these changes shouldn't happen under normal circumstances, Eddy explains. If you tried to change the meaning of a codon, you're essentially introducing simultaneous mutations all over the genome, Eddy tells The Scientist. Every place where that codon is used, you just substituted an amino acid. Part of the reason changes do happen, Shulgina explains, is that some bacterial genomes may have a low composition of certain nucleotides compared to others. That brings the usage of codons that rely on those nucleotides down to nearly zero, making it easier for an organism to survive shifts without altering too many proteins in a drastic way. At least in bacteria, it seems like these sorts of forces might explain why the genetic code evolved this way, Shulgina says. This might be totally different if we looked in other forms of life like eukaryotes. Exceptions to the standard genetic code have been found in single-celled eukaryotes such as yeast, but experts expect alternate codes to be rare in more complex eukaryotic organisms. Tracing down why these alternate genetic codes emerged during evolutionary history is difficult, multiple researchers tell the scientist, in no small part because humans couldn't watch it happen. But the authors do have some hypotheses. In one case, Shulgina identified a bacterium that uses the same alternate code as a bacteriophage virus that infects it, indicating that the bacteria seemingly evolved an alternate code that prevented its cellular machinery from being hijacked, and that the phage may have then made the same adaptatio. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.